Introduction to bacteria. So let us see what is bacteria. They are microscopic unicellular prokaryotic organisms. They do not have the membrane-bound cell organelle and they lack a true nucleus. Hence, they are grouped under the domain prokaryote together with the archaea. So they can also call the archaea, bacteria, and eukaryota. In a three-domain system, bacteria is the largest domain. That is, living beings are classified into archaea, bacteria, and eukaryota domain in the three-domain system. Bacteria is a single bacterium. This is derived from the ancient Greek word bacterion, meaning cane, as the first bacteria observed were bacilli. The study of bacteria is called bacteriology, a branch of microbiology. So here we're going to see what are bacteria and the evolution, ecology, structure, shape and size, classification, feeding and the reproduction, metabolism, factors affecting, and what are all the diseases they are causing and how we can identify them, their application and disadvantages. Here the first thing we're going to see is evolution of a bacteria. Bacteria, they are considered as the first life form to arise on the earth about 4 million years ago. All other life forms are evolved from the bacteria. A hyperthermophile of about 2.5 to 3.2 billion years ago was the ancestor of the bacteria and the archaea that are found in the present time. The endosymbiotic association between the different bacteria in 1.6 to 2.0 million years ago gave rise to the first proto-eukaryotic cell, which gradually gives rise to the eukaryotes. Ecology of the bacteria, that is, we can say the habitat. Bacteria, they evolve to adapt and survive in any kind of the ecological natures, from normal to the extreme environment. Hence, they are ubiquitous. They are found in every possible habitat on the earth, soil, air, and water. They are associated with all the biotic and the abiotic components of the earth. They are essential components of the every ecosystem. They are not only in the normal ecological habitats but are also found in the extreme environment. Such bacteria are called extremophilic bacteria. They are found in the extreme cold psychophiles and extremely hot thermophile and extreme pH acidophiles and the alkalophiles. Extreme pressure barophiles, anaerobic environment anaerobic and the acidic area xerophiles, high radiation area toxic waste and the barren on the rocks deep underground and maintain the dip etc. Soil, this is the most heavily habituated places where they constitute about nearly 0.5 percentage width of the soil mass. 1 gram of topsoil mainly contains as many as 1 billion bacterial cells. It is estimated that there are approximately 2 into 10 power 30 bacteria on the earth, but only around 2% of them are fully studied to date. Hence, there is a huge research. Gap on the diversity and the ecology of the many unknown bacterial species. Wide varieties of bacteria live in the body of all living beings, including the higher plants, animals, and even the human body. In an average human body, there are about 10 power 14 bacterial cells, while the body itself is made up of about 10 power 13 human cells. Let us see about the structure of the bacterial cells. Bacteria, they are unicellular, that is made up of the single cell. They are prokaryote and the cells are different from animal and plant cells. In general, the structure of the bacteria can be studied as external and the internal structures. The external structure of the bacteria, it included as the cell wall and the structures outside the cell wall. Flagella, the first one. In the singular form, we can call it as a flagellum. Flagella, they are long, hair-like filamentous structure of about 4 to 5 cm long, which is 0.01 to 0.03 mm in diameter. They confer the motility to the bacteria. Flagella, they are divided into 3 parts: filament, hook, and the basal body. The filament is a thread-like part extending outside the cell wall. It is made up of flagellum protein. The hook is a short curved structure that joins the filament with the basal body. It produces the repulsion like the propeller during the revolving of the flagella. The basal body is a set of rings embedded in the cell wall and the plasma membrane. It consists of two pairs in the gram-negative bacteria and one pair of rings of the gram-positive bacteria. It synthesizes the polymers of the flagellum, produces the energy of the evolution, and regulates the movements of the flagellum. Functions of the flagella. They are responsible for the motility, aids in the chemotaxis, and aids in the bacteria pathogenicity and the survival. Villi and the fibria, they are short, hollow, and the non-helical filamentous structure of about 0.5 μm in length and 0.01 μm in diameter. They are exclusively found in the gram-negative bacteria. They are composed of protein, villi, arranged non-helically. They are short, numerous, and straight than the flagella. 6 pili. They are the special kind of pili that take part in the bacterial conjugation. They are larger than the usual pili, which is 10 to 20 mm in length. They are few in number, just 1 to 4 in number. They are further classified into two types, F. pili and I. pili. The functions of pili or fimbriae, they act in the adherence to the host cell. 6 pili helps in the bacterial DNA transfer during the bacterial conjugation. Capsule. It is a viscous outermost layer surrounding the cell wall. It is composed of either polysaccharides or polypeptide of both and water. They are present only in some species of the bacteria. The capsule is of two types: macrocapsule with a thickness of 0.2 mm or more, and a microcapsule capsule with a thickness of less than 0.2 mm. Instead of viscous covering, some bacteria are surrounded by amorphous paracrystalline colloidal porine materials called its slime layer. Functions of the capsule. They aid in the adherence. They prevent from the desiccation, confer the resistance against the phagocytosis. The slime layer protects from the proteolytic enzymes. Sheath of the prosthesis. A sheath is a hollow-like tube structure enclosing the chain-forming bacteria, mostly aquatic bacteria. It provides a mechanical strength to the chain. The prosthesis. This is a semi-rigid extension of the cell wall and the plasma membrane. It increases the nutrient absorption and also helps in the adhesion. Cell wall. The cell wall is a rigid structure made up of the peptidoglycan that surrounds the plasma membrane. As an external coat, it is of 10 to 25 mm in thickness. The peptidoglycan is a cross-linked polymer of alternately repeating N-acetylmuramic acid and N-acetylglucosamine polysaccharide subunits. Based on the composition, the bacterial cell wall is classified into two types: gram-positive and the gram-negative cell wall. Gram-positive cell wall. This is a thick cell wall containing a large amount of the peptidoglycan, about 40 to 90% of the cell wall. They are arranged in the several layers. This type of cell wall also contains the acidic sugars like teichoic acid, teichouronic acid, and the neutral sugar like mannose, arabinose, rhamnose, and the glucosamine as the matrix substances. The teichoic acid, they are made of polyribose phosphate or the polyglycerol phosphate. They are the major surface antigen of the gram-positive bacteria. They are of two types: valtechnic acid and the lipotechnic acid. The teichouronic acid is the polymer of N-acetylmuronic acid or the N-gluconic acid. This type of cell wall takes up the crystal violet dye and confer the purple color of the gram-positive bacteria in the gram staining. Gram-negative bacteria. Gram-negative cell wall is a thin cell wall with a significantly less amount of the peptidoglycan. It is comparatively more complex than the gram-positive cell wall. It contains a lipoprotein, lipopolysaccharide, and the outer membrane in addition to the peptidoglycan. The lipoprotein layer is composed of bronze lipoprotein. It is embedded in the outer membrane and stabilizes the outer membrane. The outer membrane is a bilayered structure containing an inner layer resembling the plasma membrane in composition, and the outer layer is made up of lipopolysaccharide. It is rich in the variety of proteins like purine and other outer membrane proteins. Lipopolysaccharide is a complex molecule consisting of three components: lipid A, char oligosaccharide, and O polysaccharide. Lipid A is composed of phosphorylated glucosamine disaccharide, long chain fatty acid, and the hydrosomatic acid. The core oligosaccharide is composed of two sugars, ketodeoxyoctonic acid and the heptose sugar bonded together by the lipid A. O polysaccharides are composed of the wide variety of sugar that differ in between the bacterial strains. This confers the different antigenic properties to this different bacterial strain. They lose the crystal violet during the decolorization step and take up the sapphire during the counter staining, hence providing the characteristic spin color to gram-negative bacteria. Next, we can go for the cell wall of the acid-fast bacilli. It is unique with the large number of the microlic acid. They resist the decolorization of the acid alcohol or sulfuric acid, hence called the acid fast. Bacteria without the cell wall. The mycoplasma is a minute bacteria without a cell wall. They do not have the fixed shape. Besides the natural bacteria, there are several other cell walls deficient forms like a protoplast, spiroplast, and the L forms. Gram-positive bacteria and the gram-negative bacteria. They contain higher peptidoglycan content, lower peptidoglycan, lower lipid content, higher lipid content. The main components are peptidoglycan, teichonic acid, and the lipouronic acid. The main components are peptidoglycan, lipoprotein, lipopolysaccharide, and outer membrane. A very few amino acid without any aromatic amino acid. Wide variety of the amino acid with different aromatic amino acid. Internal structures of the bacteria. Internal structure of a bacteria, it includes the cell membrane and the structure of the cell membrane. Cell membrane, it is the innermost phospholipid bilayer just beneath the cell wall, enclosing the cytoplasm. It is a thin semi-permeable membrane. Unlike eukaryotic plasma membrane, they lack sterols except in the mycoplasma and comparatively have the higher proportion of proteins. In place of sterols, they have sterol-like compounds called the hapanoids. They contain a wide variety of the fatty acids like usual saturated and the unsaturated types, and additionally methyl hydroxyl or the cyclic groups too. The plasma membrane is equipped with the several porine proteins for the passive transport of nutrients and ions. Functions of the cell membrane or the plasma membrane. They are selectively permeability, which regulates the inflow and the outflow of nutrients, ions, and the metabolites. Electron transport and the oxidative phosphorylation can be done because of the plasma membrane particles. Cytoplasm. It is a colorless, colloidal, viscous fluid with suspended organic and the inorganic solute enclosed within the plasma membrane. Unlike eukaryotic cytoplasm, they lack membrane-bound organelles. They have ribosomes, lysosomes, inclusion bodies, nucleic acid floating in them. Ribosomes. The bacterial ribosome are 70S type and quite smaller than the eukaryotic 80S type. They are made up of two subunits, the 50S and 30S. The main role is to synthesize the bacteria proteins and enzymes. They are the target sites for the different antibiotics like erythromycin, macrolides, aminoglycosides, etc. Lysosomes. They are vesicular or branched structure formed by the invaginator of the plasma membrane. They represent the eukaryotic mitochondria in function and are the site of action of the bacterial respiration enzymes. Inclusion bodies. They are believed to be the storage food. They are of two types. Organic inclusion body containing the glycogen or polyhydroxybutyrate granules. Inorganic inclusion body containing the polyphosphate or the sulfur granules. Bacterial nucleus. They are called nucleoids. Unlike eukaryotic nuclei, they are not enclosed in the nuclear membrane and lack nucleus and the nuclear plasm. It is represented by the dsDNA molecule either in the closed circular form or in the coiled form. Bacterial DNA are found either in the nucleoid or as a chromosomal DNA or outside nucleus as a plasmid. Endospore of the bacteria. Some bacteria under the stress form the dominant stage called the endospore. They are produced during the unfavorable environmental condition. They grow to the vegetative form when the conditions become favorable. They have the four distinct structure of proteins: core containing nucleoid and the condensed cytoplasm, spore wall, and the important innermost wall of the peptidoglycan, cortex, thickest wall with the unusual peptidoglycan, protein coat, and outer impermeable layer, made of the keratin-like protein. Next, we can go for the shape and the arrangement of bacteria. There are four distinct types: cocci, bacilli, spiral, and comma shape. The cocci-shaped bacteria, they are spherical bacteria. Based on the arrangement of cell, they are further subgrouped as monococci, diplococci, streptococci. Monococci, which is a singular cocci; diplococci, two spherical bacteria are arranged in pairs; and streptococci, they are the spherical bacteria that are arranged in the long chain form. Next, we can go for the Staphylococci. They are the spherical bacteria that are arranged in the irregular clusters like bunch of grapes. Tetra, they are arranged in the group of four cocci. And Sacchae, they are arrangement of a cocci in the group of 8. So these are all the shape and the arrangement of the bacteria: coccus, diplococci which is encapsulated, and staphylococci form like a custus. Streptococci form a chain. Sacchae, they are made of 8, and tetrad made of 4. The same thing is followed for others in the bacillus also. Bacillus shaped bacteria, they are rod shaped bacteria. Based on the arrangement of cell, they are subgrouped as bacillus, monobacillus, diplobacillus, streptobacillus, palisade, coccobacillus. Single unattached rod-shaped bacteria. If the bacilli is arranged in pair, they are diplococci. They are arranged in chain, they are called streptobacilli. If they are fins-like form, they are palisade. And oval-shaped, that is round ends, or oval-shaped, they are coccobacilli. Spiral, they are long helical-shaped or the twisted bacteria. Comma-shaped, they are comma-like in structure. Besides their four basic shapes, several bacteria are found in the other shapes like filamentous, star-shaped, appendage rubberding, pleomorphic, Chinese-like letter, lobed, stalked, and sheeted one. Size of the bacteria. The bacteria are microscopic with a wide range of sizes from 0.7 to 100 mm. Cocci, they are generally 0.2 to 1 mm. Bacilli, 1.0 to 5 mm in length. Spirochetes, they are 20 mm in length. Smallest bacilli are the Pelagobacter ubica. Smallest cocci, Mycoplasma. Largest bacteria is Theomycariata nabuensis. Classification of the bacteria. There are different schemes on the classification of the bacteria. Some of the most common schemes of classification based on the gram staining. It is the most common mode of classification used widely in the medical and the research purposes. They are grouped into two groups: gram positive bacteria and the gram negative bacteria. Gram positive bacteria, they having the thick peptidoglycan layer and retaining the purple color of crystal violet during the gram staining or the gram positive bacteria. They are Staphylococcus, Streptococcus, Enterococcus, corn bacterium, Streptomyces, Bacillus, Haemophilus, Clostridium, Listeria. Gram negative bacteria having the thin peptidoglycan layer, losing the crystal violet but retaining the pink or red color of the counter stain, saffron during the gram staining. They are the gram negative bacteria. Example: Escherichia, Salmonella, Shigella, Neisseria, Klebsiella, Proteus, Pseudomonas, Enterobacteria, Citrobacter. Here we can go for the comparison of the gram positive and the gram negative bacteria. They are violet or purple, they are red or pink during the gram stain. Thick cell wall, thin cell wall. Thick peptidoglycan, thin peptidoglycan. Higher mucopeptide, lower mucopeptide. Low phospholipid, high phospholipid. Mesosome present, mesosome absent or rarely present. Fimbriae or pillae are absent, fimbriae or pillae are present. Form endospore, form exospore. Produce exotoxin, produce endotoxin. Tecnic acid present, tecnic acid absent. Lack on the outer layer, presence of outer layer. Classification of the bacteria based on the oxygen requirement. They are classified into three types: aerobic, anaerobic, and the facultative. Aerobic bacteria, they respire aerobically and can't survive in the anoxic environment. Example: Pseudomonas, Nocaria, Mycobacterium. Facultative aerobes, they survive in very low oxygen level and can survive both oxygenic and the non-oxygenic environment. They are microaerophils. Example: E. coli, Klebsiella, Lactobacillus, Staphylococcus. Anaerobic bacteria, they respire anaerobically and can't survive in the oxygen-rich environment. Example: Clostridium, Listeria, Campylobacteria, and the bacterioids. Classification of the bacteria based on the optimum temperature. They are classified into three types. Psychrophiles, they have the optimum growth temperature of 15 degrees Celsius or below. Example, Chrysobacterium, Psychobacterium, Spingomonas, Listeria. Mesophiles, they have the optimum growth temperature 15 to 45 degrees Celsius. Pathogenic bacteria fall in this category. Example, E. coli, Staphylococcus, Salmonella typhi, Streptococcus, Klebsiella, Pseudomonas. Thermophiles, they have the optimum growth temperature at about 45 degrees Celsius. Bacillus thermophiles, Methanotroids, Thermophilus, they all comes under this category. Classification of the bacteria based on the arrangement of flagella. Atraches, they are the bacteria without the flagella. Example, Lactobacillus and the Staphylococcus, Streptococcus. Monotraches, they are the bacteria with one flagellum and one pole. Example, Campylobacter, Vibrio cholera. Lopotraches, they are the bacteria with the multiple flagella. Example, Spirillum helicobacter. Peritraches, they are the bacteria with the multiple flagella projecting in all directions. Example, E. coli, Klebsiella, Proteus, Salmonella. Amphitraches, they are the bacteria with one flagellum at each poles. Nitrosomonas. Classification of the bacteria based on the mode of nutrition. Autotrophic, they are the bacteria capable of assimilating the inorganic matters into organic matters, capable of preparing their own food like plants. They are of two types, photoautotrophs, chemoautotrophs. They use the energy from sunlight for the assimilation. It includes cyanobacteria, purple sulfur bacteria, and the purple non-sulfur bacteria, green sulfur bacteria. Chemoautotroph, they have the chemical energy for assimilation. It includes sulfur bacteria, nitrogen bacteria, hydrogen oxidizing bacteria, and the methanotroph ion bacteria. Heterotrophic bacteria, they are the bacteria that derive the energy by consuming the organic compounds but do not convert the organic compound to the inorganic one. They are parasitic or the symbiotic type. Example, E. coli, Staphylococcus, Mycobacteria, Klebsiella pneumoniae. Saprophytic bacteria, they are the bacteria that decompose organic compounds into inorganic and derive the energy. They are decomposers and feed on the dead plants and animals. Feeding in bacteria. Bacteria feed on the several organic or the inorganic compounds. The food enters the bacteria body either by the phagocytosis or by osmosis and diffusion or through the protein channels. They obtain the energy by either the photo or the chemosynthesis, decomposing the organic compounds or breaking down the inorganic compounds. Based on the feeding habits, they are grouped as autotrophs, heterotrophs, and saprophytes. Thank you.